Hello, my name is Leah. In today's class we will be looking at the Human Genome Project. The human genome is a molecular arrangement of the chromosomes in the reproductive cells of the human body. For years doctors have been trying to figure out this blueprint for all physical and behavioral traits that an individual can pass to their offspring. Essentially, the human genome is the code for building a human being. This mapping of human anatomy has developed substantially over the years with the aid of new technology. Mendel's statistical laws are today the foundation of modern science of genetics, thus the creation of the Human Genome Project began. There was this belief that we could answer huge amounts of things just by studying genes and gene variants, states geneticist Tim Spector of King's College London. Yet 26 years in the future this prospect seems more distant than ever. In the 1990s, the US government sponsored a project to decode the sequence of 3 billion chemical base pairs in all human genome by issuing grants and funding research programs. Later it became an international effort under the leadership of American molecular biologist James Watson and later Francis Collins. Once the genome was sequenced, the next project was to attempt to understand specific parts of the genome. In an attempt to fully understand the entire blueprint for the human body, scientists concluded that by decoding it all will help them understand how human organisms worked. Not only does this DNA aid us in understanding humans, it also helps us elucidate the relationship between humans and other animals. The goals of the Human Genome Project are to identify all the estimated 80,000 genes in the human DNA, determine the sequences of the 3 billion DNA chemical bases, store this information in a database, develop tools for data analysis, address the possible ethical, legal and social issues in regards to the HGP. The first gene was decoded in 1975 from a DNA virus. 2 years later scientists were able to create a gene that isolates a blood protein. An English chemist by the name of Frederick Sanger invented the chain termination method in the hopes of decoding DNA in the 1960s. This method was based on the careful combination of chain-terminating dideoxynucleotides by DNA polymerase in vitro DNA replication. In the 1960s the simplistic image of our DNA emerged. It consisted of proteins, and the double helix could be unzipped to allow RNA copies of these recipes to be made and be sent to the protein-making factories in the cell. Later in the 1980s Michael Hunker-Piller, aided by other scientists and engineers at Applied Biosystems, developed a new method using fluorescent dyes to locate genes that made up sequencing much faster. Scientists worked with the DNA of simpler organisms such as bacteria to determine a preliminary idea if their sequencing method worked properly. Results showed recurring strands of gene sequences, called transposons, that were like the repeated phrases in a coded message that could be used to help decode it, creating a stable resource or library of DNA clones. Assembling the larger sequences from fragments required sophisticated computer analysis. Scientist James Webber and Gene Myers theorized that large statistical samples could be used in what came to be known as the shotgun approach to researching the genome. Computers allowed scientists to multitask by matching sequences from all the genes at once. In the 1970s the first production of recombinant DNA molecules, using enzymes occurred. Recombination means crossing over. The further the two genes are apart, crossover and non-crossover events occur equally. The closer, the less crossover events occur. Recombinant DNA is a technology that involves joining the DNA from two different species and successively inserting the crossbreed DNA into a host cell. The steps of recombinant DNA include cutting and joining the DNA, monitoring the cutting and joining, amplifying the DNA sequence using a polymerase chain reaction, and finally transforming the host with the recombinant DNA to allow genes to be carried into nuclear DNA in multicellular organisms. This means that nearly all of the genome is in play for doing something, or if you change it maybe it would have an effect on something somewhere. To determine the human genome, scientists first mapped and characterized chromosomes, known as physical mapping. Sequencing followed to determine the order of DNA based on a chromosome. This is known as genetic mapping. Researchers use genetic linkage for genetic mapping. There are two different types of maps. Maps that measure the distance between two genes in regards to nucleotides, and genetic linkage maps. When scientists discover a new mutation, They look for a linkage to other genes to determine the location of the mutation. Although the process of identifying genes is too complex to be achieved by studying linkage maps, due to the large variety of DNA. Thus working with DNA and looking for a particular gene is difficult. Of the 1,000 or so possible genes on chromosome 22, only 545 are easily to spot. Linkage maps are determined by studying percentage chances of linked genes staying together or separating in the process of crossing over. Therefore, linkage mapping does not convey an accurate picture of specific genes on a chromosome. This type of mapping only shows the relative distance between genes, not used for identifying characteristics like inherited diseases. Overall, the Human Genome Project is considered to be a major success in the science world, delivering a 99.9% completed rough draft of the human genome sequence in 2000 and later being completed ahead of schedule and under budget in 2003. The medical and science visionaries who poured hours and months into this project could clearly see how genomics would ultimately advance medicine. Genetic findings are guiding treatments for 4,000 common diseases like the blood thinner clopidogrel, which is prescribed to prevent platelets from binding inappropriately, thus causing strokes. There have been multiple discoveries of rare genetic mutations. While there are multiple examples of genetic analyses providing much-needed answers or new therapeutic approaches in the medical field, limitations of the data obtained is still an issue. Research remains to be done to ensure a productive incorporation of genomics into clinical care. DNA makes RNA, which makes proteins, provides only a simple sample of the relationship between genes and organisms. Biochemical options form a complex network that is not only reducing life to a series of genes located on chromosomes. There is a need to still understand how cells' many components work together. Knowing molecular sequences, the order of genes, is not the same thing as knowing what each gene does or what the trait is responsible for. This requires a complex investigative process of elimination those sequences that are just noise. Until scientists know which molecular sequence is responsible for a particular trait, we cannot design drugs to reverse the effects of genetically caused diseases or deformities. In some cases, genes contain non-coding regions that regulate gene expression. Introns is a region which is removed from the messenger RNA in a process called splicing. Depending on how it is spliced, genes may code for different proteins. Genetic coding is interconnected with coding of other genes. All are codependent. They will need to map and edit. The human genome is a long way from being a perfectly honed, finished project. Rather, it has been crudely stitched together, and is the product of crazy uncontrolled experimentations that would be rejected out of hand by the ethics board. And the process is still continued till this day. Thanks to embryo screening, scientists are beginning to take control of human evolution and thus an era